Good afternoon, everyone. Thanks for joining us in person and to you two on Zoom. What a fantastic day. And we get to hear from a phenomenal panel of people with a wealth of expertise spanning a range of industries. But before we start, and on behalf of TRI, I wanted to pay respects to the Turrbal and Jurgara peoples, the traditional custodians of the lands and waters of the Brisbane area. We recognise their continuing connection to land, water and culture. We pay respect to elders past, present and emerging. We acknowledge the stories, traditions and living cultures of Aboriginal and Torres Strait Islander peoples and commit to fostering a culture of learning from and working with First Nations people in spirit of reconciliation and access to justice. Well, again, thanks very much everyone for joining us today. And for our next seminar series, which is engaging with an investor and pharma. And we've got a phenomenal panel that are presenting today. We have Professor Anand Gautam from Pfizer, Dr. Adriana Basmarelli from CSL, who's joining us online by Zoom. Unfortunately, we can't hear from Dr. Goz Shepherd today, who's unwell at these times. So that enables everyone else a few extra minutes to present. We also have Dr. Craig Belcher from UniQuest. We have Associate Professor Samira Hasnain from our very own TRI here at from the MARTA. And as well as that, we have Dr. Rosalind Wilson, CEO of Carpe Vitae Pharmaceuticals. So it's fantastic to have you all. So how it's going to work, we have three speakers now, and then our two panelists who we'll invite up at the end. And we've got time for questions from the room and online. But to kick things off, I'll just invite Dr. Anand Gautam up to present. So just quickly to tell you a little bit about him, he's the Executive Director and Emerging Science Lead from the Emerging Science and Innovation Pfizer, in charge of Australia, New Zealand, Southeast Asia. He's a wide-ranging leadership roles in R&D and innovation strategies in inflammation, autoimmune diseases, cancer, at the international biotech and pharmaceutical companies for the past 30 years. Before joining Pfizer in 2017, Anand was a Senior Director of R&D Innovation Sourcing at Novo Nordisk, Copenhagen, and VP of R&D at Progen from here in Brisbane. As well as that, he was a SVP of External Science and Collaboration at Pharmexa in Denmark. Thanks very much, Anand. Share your screen.
My screen. Oh no, where is that? So
yep. No. Screen share.
Share. This one. Can I just use this, right?
Yep.
To play.
Yep.
Full screen.
Yep.
Okay.
Oh, actually, yeah, that one. Yeah.
How come I'm seeing myself? That's okay?
Yeah. That's the one there.
Okay. Put this one away. I don't want to see myself. Yeah. Well, thank you for inviting me here, and look forward to presenting this quite challenging task as to how to attract investors and pharma to do business with institutes like TRI. And some of you may have heard me before, so I'm trying to differentiate a little bit. So hopefully you haven't heard the same wordings, because I was here about 5-6 months ago and I gave a 1-hour talk on that one. So I then talked about meaningful innovation, which is still the same. You know, when we do some research, it has to have some meaning. It means that it basically needs to help the patient. I will stop there because I think I talked a lot about what other means is. Because patient really doesn't care about Nature Cell science paper, patient care about treatment. And when you work with big pharma, whether it's Novo Nordisk or Pfizer, our exit is not return on investment. Our exit is when we take the drug to the market. So it's a very, very different mindset. And that's why it's actually fun to work with large pharma, because we are looking at the real end game as to what patient needs, which is a drug, not a science paper. So let's see how we can do partnerships. So I'll just briefly describe what emerging science and innovation function is at Pfizer. And then I would touch upon a touchy issue, which is called how do we in Australia become globally competitive. So what patients really demanding now is not just prevention and remission, they're actually looking for cure. So we need to be able to find quality of life for patient that is like us. They should be able to walk around with quality of life, really important for all of those patients who suffer from diseases. And that's where Pfizer is actually focusing on. Of course, we're looking for prevention. Of course, we're looking for remission, but long-term aim is to find ideas that can delay the disease so long that these guys live like they've been cured. So that's our ambition. So what are the key factors for novel drugs that we look at it? I think some of you may have seen this slide before, so I apologize, but I think the message needs to come through very loud and clear. That is, we're looking for these 3 Ds. If I was in a long talk, I'll ask what these 3 Ds were. But basically, we're looking for differentiation. We're looking for what the disease landscape will be in 5 to 10 years' time when your ideas may become a drug. And that's very important. It means that we scan all late preclinical to phase 3 programs that are publicly available on the data sets. And if there are 100 clinical trials going on breast cancer, then we say, well, if I get the drug from you, where would that fit in 10, 12 years' time? And that's very important, even though the science may be superlative. So we look at disease landscape 10 years down the road when your ideas might become a drug. And at that point, what the demand will be, because we expect people to be healthier in 10 years' time. There should be more treatments for diseases. So what the demand will be. So this is basically our metrics that we focus on: differentiation, disease landscape, and demand. So whenever I come to TRI or other institutes, I actually ask that question: show me the differentiation. And that's number 1. And of course, that's where the innovative partnerships happen. And there are so many ideas currently here and other places, so much substrate that we can look at and working with. And I'll give you a few examples of some of the collaborations that we've set up in Australia and also overseas in my area. So I'm actually now responsible for the whole of Asia, not just Southeast Asia. So I look at China, Japan, Korea as well. And it's been fantastic to work and being reminded as to why we do partnerships. And we can have these discussions on these points at the question and answer session. This is just one plug on Pfizer, but I think you should be looking at any pharma industry, not just Pfizer, in terms of collaboration, of course. But this is just to show what Pfizer's current thinking is in terms of where we're going. On the top, it shows the disease areas that we work on: rare diseases, mainly monogenic diseases, oncology, immunology, inflammation, basically autoimmune diseases, internal medicine being cardiovascular effect, cachexia, obesity, even diabetes, that should be there. Vaccines, of course, you know about. And at the bottom of each of these, at the bottom here, is basically talking about the current areas of unmet wish list that we have, which is repeat expansion diseases, where you might have repeats of nucleotides and that causes disease, or DNA damage response, or cellular senescence. How can we make a cell go to sleep in cancer? How can we make the cell wake up in cancer in terms of inflammation, or making T cells do a good job, not die? And the other thing that we have recently incorporated, which we're working on, and there was a call for proposal for RNA, is how we can use RNA technology, mainly mRNA technology, as therapeutics. And I was surprised that there were about 60, 70 proposals worldwide that came for CTI at the recent call for proposal. And I was very pleased to have 7 or 8 of them from Australia. It means that there's still a good substrate, at least the initial look that we did. Of course, we don't know how many of them will progress further, but at least in the initial screen, there were 7 or 8 that kind of sounded okay. So it's also always important when you want to do partnering to have a bit of a look at what the company is really looking for at present time. That's the message. So rather than just going to J&J and say, look, I've got an idea. If you had a bit more information like this, then you can say, look, I'm working on any of these modalities or any of these sciences, biologies that could be useful. That's very, very important because sometimes I get emails which are extremely annoying about repurposing and so on. Of course, they are important areas, but this is not the current strategy for Pfizer. So I think in most cases, if you had done some homework, it's always useful for pharma to then be more receptive to your ideas. And that was the same at Novo Nordisk when I was there for 8 years. So each company, each pharma has a way of sourcing innovation. At Pfizer, we have these emerging science leads, which is me, and we are about 15 of us across the world. I cover Asia, similarly Europe and United States. Each company has a way of partnering, what we call Center for Therapeutic Innovation. Here we're looking at science where you may have a target already, and then Pfizer can come and screen or make antibodies, etc., and then take it further. And that target needs to be differentiated. I want to repeat those 3 Ds, and that all goes into evaluating when we look at targets and CTI. Even earlier than that, we have something called target science. So we have collaborations in Australia, such as Laura Mackay and others, where they're looking at targets in areas that they lead. And that means the target may be found, but the biology is already so good about, say, tissue-resident immunity or whatever that Laura works on, that we got excited, we funding her and hoping that there'll be 1 or 2 targets coming out of it. That's even before CTI. And of course, we have Pfizer Venture. It means that if we have a VC who wants to fund research, we can be syndicate. And of course, licensing comes on top. So we can always license a molecule that is already developed, but that doesn't usually happen at university setting. Usually it's in early biotech that we can license a molecule that is targeting a novel target. The bottom line is that emerging science means that we're looking for emerging science. We're not looking at past science. And usually I for one get excited when I have not heard about a target. If I had heard about a target, that's dampener. It means that you are not the originator of the target, you are a follower of the target. So we're looking for those investigators that are driving the field, finding a new target. And that's the way. It doesn't mean that following a target is a bad science. It's just that this is the current demand for emerging science, because why should we invest money on known target while the other guys are also working on it? So I'm looking for truly novel innovation, but other companies have different requirements. So don't just take Pfizer as this is our current strategy. This is just to give you an example that I've not been sitting and talking like this for the last 5 years. There have been numerous collaborations, including licensing deal at the top with CRC for Cancer Therapeutics, which is basically a conglomerate or consortia of multiple universities. And they had a molecule targeting KAT6A. Pfizer was very interested in the molecule and the target. And so we licensed it with $20 million up front. And there already been milestone paid for that. There's been a lead milestone paid. There's already been a phase 1 initiation milestone paid. And that's an extremely satisfying, personally, because we were able to license that from Australia. First real license from Australia. And then there's a list of many investigators, not named, except Laura and maybe 1 or 2 others may have been, that we announced. Chris Miney, one in St. Vincent, and Mark Dawson and Peter McCallum Institute that we announced. But there are, and so many examples of where we're funding top science in Australia at the moment. In addition, you may have heard, it's not a finished deal yet, but Pfizer also made an offer to ResApp, which is, you can say, an algorithm-driven app-based company here in Brisbane for $100 million about 2 weeks ago. Of course, that requires shareholders' approval, but $100 million by Pfizer in Brisbane to a company that very few people knew came from UQ, which had very little investment from local investors. And Pfizer found their algorithm interesting, and we put $100 million on the table. Even if that deal doesn't go through, we have licensed their COVID-19 algorithm to do work with it. Either way, it's a win situation. So it just shows that how much money just in the last 5 years I'm not claiming myself, but I've been here 5 years. We put close to $150 million in Australian science. And I think that just shows that you could be a small shop, but so differentiated that we can be interested in licensing. And currently I'm looking at many different opportunities within Queensland, of course, Australia-wide as well, that are quite differentiated. So it's been a fantastic 5 years with Pfizer. This is just to put a small seed of why we fail in Australia. Sorry to say that. This is something we got what we can do better in Australia going forward. This is a list of drugs that have been approved by the FDA over the last 25 years. And there's like 780 or so drugs been approved. If you put Australia on it, we talk about Gardasil as the drug. But apart from that, we've got drugs that were approved a long time ago. And of course, Mark Sullivan's work is remarkable for river blindness. And we have got venetoclax. And this is what we talk about. I think we expect more than that. It means that overall, we're producing less than 1% of the drugs approved by the FDA. In fact, it's much, much less. This is just the polite way of saying we are very bad in translation. And this is only therapeutics I'm talking about. So please don't take me as a med device or anything. But this is a challenge we need to fix in Australia. And I know my time is running out. So we will just go through saying that there's plenty of funding in Australia. No question about that. There's a huge mess up as to who manages it in Australia. So when people say there's no funding, I argue, no, it's not true. There's plenty of money. In fact, the money that is going to our government is sponsored investment funds, I call them. I don't call them VC anymore. I call them government sponsored investment fund. So majority of funding that our investors have is government funding or the superannuation funding, but they live like VCs. So I think we need to be very clear that they are not the same VCs in Boston and San Diego. They are government sponsored investment funds. And we need to demand as to why our biotech industry is failing despite having 400 biotech companies in Australia and so on. Okay, so I think that's probably enough for me to say, except to say that, you know, academia works on early stage and then comes the pharma. And then we do deals at various stages. These are just two examples of it. And I think I'll stop it there. Thank you.
Fantastic. Thanks very much for your insight there, Anand. Now on to our second speaker. We have Dr. Adriana Basmarelli joining us from Zoom. She's a Senior Director of Therapeutic Area Lead for Transplant, CSL. Now, just to give you a bit of a background for Dr. Basmarelli, she's been in this role since 2020 and has been worked on developing the strategy for transplant, expanding the network of collaborators and developing a portfolio of projects to support a leadership role for CSL in transplantation. Dr. Basmarelli has worked at CSL since 2008, where she has taken on such roles as the leader of the adjuvant and vaccine team in CSL research, contributing to several vaccine projects, as well as a director of in vivo biology. And prior to this, Dr. Basmarelli was actively involved in research and teaching in Latin America, Europe, and more recently here in Australia from 2005 onwards on NHMRC funded research. Okay, we'll hand over to you, Adriana.
Thanks, Mark. Let's see if I share the screen. Let me know when you can see it. Hopefully the presentation worked. Can you all see it?
Yep, all good.
Excellent. All right, well, thanks for the invitation, Mark, and also thanks to Ranjani. I'm really excited to be here talking about what we love to do more in CSL, which is bringing new drugs to patients and collaboration. So I would like to extend a hi to all my friends over there in Queensland and my current collaborators. So I would like to start by sharing with you what are the CSL's areas of interest for collaboration in a broad sense. So from the point of view of platform development, CSL is committed to three main platforms for drug development. One is in cell and gene therapy, using lentivirus delivery as a platform. And then we have, as you know, a long history of plasma fractionation for protein therapeutics, as well as recombinant technologies, platforms for developing recombinant proteins for therapies for different diseases. And this involves protein engineering as well as monoclonal antibody production. So if you have a project or an idea that will help us to improve these platforms, we would love to hear about it. And there on the bullet points on the sides, there are some of the examples where we could potentially look for external collaborations. When we prepare our data package to decide on whether to develop a drug or not, we utilize a lot of different tools, particularly animal models that are relevant for disease, for human disease, and they are translatable and very well validated. So this is an area where we have several collaborators in allowing us or enabling us to access to those tools. In addition, human in vitro models that can mimic some of the processes or pathology pathways related to human disease, that's something we are interested in as well. Biomarkers is at the key of our attention. Definitely they are important for understanding pathophysiology of disease, as well for the stratification of patients in diseases that are heterogeneous, and there are patients with different disease phenotypes, as well as to determine response to treatment. So these are very key areas where we also have some collaborations externally in this. And then when we come to targets, we are looking for novel targets to treat different diseases, and I will touch on that in the next slide. And in terms of the modality of those therapeutics, they need to be aligned with our internal platform. So if you have a particular target that you can modulate with an antibody, a protein therapeutic, or cell or gene therapy, you are very well fitted to collaborate with CSL. In more details, CSL is working on a model of therapeutic areas, and these are our 5 core therapeutic areas: immunology, hematology, respiratory, cardiovascular, metabolic, and transplant. So we have one lead in research for each of these therapeutic areas. I am representing the transplant, and we support the development of drugs in these therapeutic areas using, as I said before, our 3 core platforms. Now, for each of these therapeutic areas, every year we review our strategy and our indication focus. And there's a list of indications associated with each of these therapeutic areas where we are interested in developing drugs. And that can be disclosed at the time that you reach out to us with the intention of establishing a collaboration with us. More specifically, what we are looking in the data package. So we are very focused on the value and the strength of the scientific data package. And there in red are the highlight of what we will be looking in your proposal or in your project. So first we want to understand what's the rationale for having chosen a particular target and how much you understand around the target biology. So we can understand how is that, how your data really support going after that target for a particular disease. We are looking also at the safety. So we're interested in understanding from your data where that target is being expressed and whether you have really demonstrated any potential off-target effects that could compromise the safety of and utility of that particular therapeutic. Moving into the interactions, in many cases we have proposals where we have a therapeutic, but the ligand or the target is not very well clear. Well, that's something we would like to understand. We really want to understand how your molecule works, even at the molecular level. And also if you understand very well, which is the ligand for your therapeutic. We want to understand polymorphisms. We want to understand translatability of that particular target. So we can have a very good understanding of the utility and applicability of that particular therapeutic. And efficacy in vivo proof of mechanisms are key components of the data package that we would like to see. So we want to understand that your therapeutic works and in which disease, And you can do that using, as I said, translatable or validated animal models. We want to understand, to see from your data whether you know how your molecule works. Not only you see an effect and a modification of the disease, but you know exactly which pathways and what your therapeutic is modifying. And more importantly is how your therapeutic compares to the current standard of care or to any other drugs that are in development that might reach the market before your drug does it. And this is the benchmarking, and this is also very important. And finally, the target validation. So we really need to understand that your data is translatable. So, and that target is really linked to a human disease. And the modification of that pathway has a high probability of success in modifying the disease. And of course, we value the track record and the expertise of the academic team around that target, that area of biology of that particular disease. So this is my last slide to keep on time. So if we show you in a broader sense what an optimal data package will look like is, first, you really understand what the unmet need is that you're trying to target and to address. And your therapy really addresses that unmet need. Either you have validated the target in the human disease, you know that inhibiting that pathway is going to have an effect on the disease because that pathway is central to the pathophysiology, and you understand the disease in the human population. You know, for instance, that there will be a clinical development pathway. You know how many patients are there, you know how homogeneous the disease is, and if it is not, there is a set of biomarkers that will allow us to stratify the patients and define the target population. I've already touched around proof of efficacy and proof of mechanisms. This is really important to know that your therapeutic is going to work in a particular disease, that you understand the mechanism of action, and you have a good benchmark data set to allow us to position your drug in the competitive landscape and in the current standard of care. Safety, as I said, off-target effects and also immunogenicity. So if you can do in silico analysis for immunogenicity, that will be really important. This is a question that keeps coming continuously, particularly when we're talking about recombinant modalities. And these last two boxes are also something we value. I mean, we need to have a good understanding of what your intellectual property position is and where is the freedom to operate with if we were going to develop your drug into the clinic for a particular indication. In terms of the competitive landscape, you've heard in the previous presentation, differentiation is key. So, and the differentiation of your therapeutic has to be really very strong, not just from the current standard of care, but also from any drugs that is in the pipeline that, as I said, might reach the market before your drug. Any other advantages, and I listed some of them there, that could be studied in vitro or in vivo models is also very important. And finally, if you have done any developability investigation studies to understand how scalable, how stable your processes are, that would be really very important for us to discuss with us. So at the end, how and when to reach out, as soon as you know that you have a therapeutic and a drug that is a good target, that aligns with our platforms and aligns with our therapeutic areas, I think that's a good time to reach to us. And as I said, we are really looking for many more collaborations than the one we currently have. And I think I'll leave it there. Thanks so much for listening.
Right. Thank you very much, Adriana. Okay, so after our third speaker, we'll just invite Dr. Craig Belcher up, who's the Senior Director and Head of Life Sciences, UniQuest. Dr. Belcher has over 20 years of experience in commercializing technology within the biotechnology, nanotechnology, and advanced materials industries. Dr. Belcher joined UniQuest in 2004 as the Manager for Innovation and Commercialization Development, responsible for a variety of institutes across UQ. And in this role, he closed over 15 deals, bringing in $50 million of research income to the university. Okay, we'll hand it over.
Very good. Thanks, Mark. Thanks, everyone. Thanks for the invitation to present today. So look, I am 3 or 4 speakers before you get to your panel. Probably just as well that Goss couldn't make it because there's going to be a bit of repetition. I will come from a different perspective though, and that is if you think about it in the value chain, academic research moves through into translational research. And then as per slides you've seen from Anand and Adriana from CSL, that moves into a biotech and then ultimately into pharma, into patients. So I am more like yourselves in the university sector looking to work up the data package such that there may be a company like Pfizer or CSL or a venture capital if Goss was here to actually take on that technology, pick up the mantle, work with you further so that it can go into their organisation and ultimately to address and help patients. So my name is Craig Belcher, as mentioned, Senior Director and Head of Life Sciences at UniQuest, PhD from UQ and worked in industry, particularly several years in the UK with an antibody company called Cambridge Antibody Technology. So this is the value chain that I mapped out, just trying to keep it super high level. This, in a very simplistic way, is the way that intellectual property or your smarts gets progressed through to something that is a product, ideally. And that product to succeed today has to have certain features to win in the marketplace. So that's what's ahead of you. As for this institute, TRI and all the people contained in here and your own respective research institutions, you know, you're in that D3, D4 space, really into translation. Now, here's what I'd like to provoke. Think with the end in mind. Who would want what you have if it worked? Let yourself think about it. If your science worked, what would they want and why? And then how do you guide your research towards that? So we have heard from pharma participants in the value chain today. You've heard the word differentiated a few times. That is more than just different. That is about being better than the standard of care by the time that your science, if it becomes a product, gets to market, which will be several years, if not a decade. So that is the competition you need to peg yourself against and to show why you're truly differentiated. So that has to be a high level of innovation and why you might win and take market share off that competition. Secondly, it is about the patients, and importantly, it's around the payers. So we hear about these very expensive drugs, but then how much you might pay for it through PBS, through the payback if it's going to be reimbursed. So it has to have a cost benefit to society. And if it doesn't have that, then the pharma company may not get behind it to take it to market. Your best way to have a chance of it getting reimbursement is to have that significant differentiation in the first place. So the point is you're pegging yourself and your program to standard of care on the basis of what would be standard of care by the time you got to market, not just today. So if that's the end in mind, what does it actually mean for you? So look, the next slide really is a collation of what you've already heard. Now, again, I'm in a similar place to you. We're in the value chain together from the university side, trying to take our science and make it attractive for industry, whether that's venture or pharma or biotech to pick up. So you need to have line of sight to a new medicine. You may not have all the answers, but actual fact, to get pharma interest, you need to have a lot of the answers or a lot of the ways to remove the risk in front of you. And a key step is knowing what risks are in front of you. So do you have a target? Is the mechanism of action known? Is there a high unmet need in the marketplace? Let yourself believe the drug works. So in that ideal scenario, the drug works. How much is that point of differentiation? Is it significant enough that pharma companies will get involved sooner rather than later? Or you might need a venture capital to carry you some of the way to get a pharma interest. So the proof of concept data, yes, we see experiments that one of the easiest things you can do back in the lab include standard of care as a positive control in your experiments. So let's see how good it is against at least today's standard of care. Do use industry relevant animal models, not just the animal model that's a friend has in the same institution because it's the same disease. Actually look, ask your colleagues, your tech transfer office is part of that. UniQuest is part of that for UQ, but that information's out there. Saw a smile, so hopefully that'll be mentioned later by Samara. So get the right data in terms of, does this have human relevance? It doesn't have to be your own data, but you will really need to show that if this thing worked in your animal models, that it won't translate into humans. So use what else is out there, GWAS, whatever you can get your hands on, do some straightforward tissue analysis, some sort of expression analysis, something to prove that it's going to have a disease relevance in humans. Obviously, this whole talk is around therapeutics. I'll continue in that theme. You may have a novel target, but is it druggable? And by what mode of action? In an ideal world, from a pharma point of view, you'd have 2 to 3 hit series that actually pharmacologically modulate that target to get the phenotype or disease intervention that you're looking for. That isn't ideal, doesn't always happen. You have to at least have the target to get venture capital interest, would be my personal rule of thumb. You would need to have some of those modulators really to get pharma interest. Although, as you heard from Anand, there are programs that are coming much earlier from pharma companies. Venture capital probably won't go any earlier. Some of the pharmaceutical companies will. There will be heaps of questions you can't answer. So that's where your research plan comes into play, and then the design of that research plan, how to de-risk, take out some of the actual issues, address the unknowns that are knowable, and do that with increasingly larger amounts of money, i.e. start with small amounts of money expenditure. And if you can design an experiment like that, that makes it more palatable, makes you more attractive to a venture capitalist or a pharma partner. And again, you may not have that. We're not necessarily expecting you to have that from a UniQuest point of view. We're part of the UQ researcher team. So you come and chat with UniQuest if you're UQ, chat with your tech transfer colleagues if you're from one of the other research institutions. IP can follow. You don't have to have a patent. You have to be careful how you talk about your program. In fact, it's more attractive to have line of sight to a patent position, but not have filed yet to maximize your patent life. And in fact, see what your partner might bring into the mix. But trying to actually be fairly forthright in this conversation, given I had 5 minutes, pleased that this overlaps with a lot of what had been said earlier. So happy that there'll be questions later. And this is probably more the highlights of what you would need to think about having in your toolkit, figuring out before you engage with industry, what you would say under a non-confidential basis, what would you hold back from, so that if you get interest, then you can get under a confidentiality and then more can be shared. Adriana said, speak early. I think that is absolutely true. Venture capital, pharma want to hear early. I would encourage you, as I would from a tech transfer position in that value chain, chat with your tech transfer office first, early and first. Because if there is something to this, if there is something patentable, then know that, figure out how to communicate without destroying your patent position, but also how to actually construct your science in a way that's more amenable to what companies like this, like pharma, Pfizer, CSL, what they would ordinarily be looking for. And that helps increase your chances of success. What I've done here, I'm not going to talk through this. I'm not going to labour through it. I did mention it to Anand and Adriana, you'll be fine too. So this is a bit of a looking insight into what these 3 organisations were presenting today, what they ask, and there's a lot of convergence. This is in their public domain, so CSL, Research Accelerator Initiative, Pfizer, CTI, which we heard from Anand, and Brandon Biocatalyst, formerly MRCF, what they're often looking for. So you can see how it all converges. It's about getting the right data pack because you are actually trying to sell your program to a willing buyer. That buyer has a lot of people around the world trying to get their attention. They can pick and choose. They will cherry pick. For yourselves, you can't pivot your research direction that quickly. You can't change entirely the disease area you're working on. There is a lot of momentum in your research career. The key, therefore, is to find out how to take what you have, find who may be the right partners, and then to put it in the best light. How to find who are the right partners. You heard about these therapeutic areas of interest. Just search for that phrase, Pfizer. Search for that phrase, CSL. It will come up. Just look at who are the top 20 pharma companies. You can see who would align with your research. If your research, that line of sight plays out and works. And that's your targets, and then how to put the information forward. That's the sort of information you've heard today. And don't do it alone. You have your tech transfer professionals around you in your own institutions, so please use them. And for those UQ, MARTA, come and chat with UniQuest. Thanks very much. I'll leave it there. Thank you.
Thanks very much, Craig. Okay, so that concludes the speakers that we had planned for the day. Next, we've got two highly experienced panelists that we'd, first of which we'll invite Associate Professor Samir Hasnain up on stage. Associate Professor Hasnain leads the immunopathology group at the MARTA Research Institute UQ here at TRI. And she's been recently been awarded an Andrew Massey Career Development Fellowship and is an assistant professor. And she has recently quite a bit of experience now in commercialising her research findings. So we look forward to hearing your perspective, Samira.
Thank you, Mark, and thank you for inviting me to speak today. So most of you have probably heard me speak about this before, but I was just going to give a general overview of how we sort of got into the commercialization. It's pretty much reiterate what everyone has said today. So looking at the market size, so I've been working on non-alcoholic fatty liver disease, or more recently known as metabolic associated steatohepatitis. And this is a multi-billion-dollar market. And so the costs are estimated to at least 35 billion in the next 5 years. And the prevalence is increasing, affects up to 25% of the population. And so our target is improving lipid accumulation in the liver and inflammation in the liver, and also fibrosis. And I'll talk a little bit about that in a bit. So our strategy was really to look at the market side, but also whether there's a need for a therapeutic. So there is definitely a need, because there's no therapeutics that have been approved for fatty liver disease and NASH specifically. And so the usual treatment and first line of treatment that you get is you told by the clinician to lose 3% of your body weight, which is a hard ask, especially because more than 50% of these people actually have type 2 diabetes and obesity as well. So there's lots of off-label treatments, but they're not very effective. And so this is looking at the pipeline. So a couple of people mentioned already, looking at the pipeline, looking at what your comparators are going to be in your models. And so if you look at the pipeline, I'm not expecting anyone to read this, but these are all the drugs that are currently in phase 2 and 3 clinical trials for NASH. So this is quite scary, because there's a lot of drugs, but most of them actually fail phase 3 clinical trials. And this is why there's a massive need for it. But some of the reasons in the field are thought to be that they only target one of the pathways that are activated in NASH. So these are all the targets that I've outlined here, and the drugs that target these specific targets. So it's thought to be better if a single therapeutic can actually target multiple pathways that are activated in NASH, would have more of an efficacy and also work in this heterogeneous disease. So our basic discovery was around the cytokine interleukin-22, and we found that it improves hyperglycemia and obesity and type 2 diabetes. And from there, we also know that it improved fatty liver, and steatosis generally in the liver and metabolic disease. So at that time, we weren't really looking at commercialization. We knew that IL-22 and systemic delivery of IL-22 can actually lead to off-target effects like skin lesions and susceptibility to hyperproliferation in the gut. But there were a few long circulating IL-22 versions of IL-22 already in the clinic, which are being trialed in inflammatory bowel disease, but also alcoholic hepatitis. So we thought this was an interesting area because it targeted a lot of the different pathways that are activated in NASH. And this is why we thought maybe rather than giving systemically a dose of IL-22 or long circulating IL-22, it would be good to target it to the tissue that we wanted to be targeted to. So that would be the pancreas to improve the hyperglycemia, and in the liver to reduce the fat accumulated in the liver. So our strategy was to link it to a ligand that can target it to these two specific tissues. And so we got an NHMRC development grant to do this. And we've had a lead therapeutic candidate for a couple of years now, and investment from IP Group, VC. And we formed a company called Jetro Therapeutics, and we're leading this candidate. So of course, for IP purposes, I'm not allowed to tell you what this actually is. So I'm just going to call it Jetro 06. And so we had to provide a solid data pack. As everyone said, this is really important if you want to engage with pharma. And we showed them that we can actually target this IL-22 to specifically the liver and the pancreas. And it can be prevented from targeting the intestine, which is really important because it can cause hyperproliferation in the intestine. And also we've done some tox work where we've shown that even a large bolus dose of this targeted IL-22 can prevent targeting to the skin and also the gut. So this is really important in differentiating us from the long circulating IL-22s that are already in the clinic. And so this is my last slide, just to show that we are trying to make a really good solid data pack where we are trying to differentiate ourselves from the current therapeutics. So we're also using semaglutide, which is a diabetic drug also used in the clinic, and gaining evidence for IL-22 and our targeted version in acting on all these different pathways. So I'm just going to quickly say that we've already shown that this targeted version can reduce hyperglycemia and insulin tolerance, which is metabolically a challenge for these patients. We also show that it can reduce inflammation, also cellular stress. We show that it can improve the lipid accumulation in the livers. And more recently, after engaging with pharma, we have started doing some industry standard models for fibrosis. And we've shown in experiments that our version can actually reduce fibrosis really effectively. So I'd just like to end by thanking everyone that's actually done the work, and especially our funding bodies, in particular IP Group and NHMRC and Jetro Therapeutics for funding this research, and obviously UniQuest for helping us along the way. And thank you.
Thanks very much, Samira, for sharing your data package. Just with the remaining minutes that we have available, I'll invite Roz up, as well as the other speakers and panelists to pop up in front. So our last panelist today is Dr. Roz Wilson, who has a unique perspective as CEO for Carpe Vitae Pharmaceuticals. Dr. Wilson has 20 years experience in new medicine development and commercialization. And in this time, she's held numerous roles at Roche, as a business manager for Nucleus X, CEO for Factor Therapeutics Limited, and global head of drug development for Telix Pharmaceuticals. Thanks very much, Roz. We'll invite you for your perspectives.
Thanks very much, Mark. Is this going to be up? No, microphone for everybody online. Great, thank you. I don't have slides, but I did make a few notes as I was listening to the other speakers. Just very briefly to introduce myself. So I studied medicine in Victoria. I'm a Monash graduate from a long time ago. It's tragic to think that I've got more than 20 years experience now, but it happens to the best of us. And then I was lucky enough to get a job in Big Pharma. I started working for Roche, or Roche as they're called here. And then I moved to Europe, and I worked with them in Europe, in the UK and in Switzerland for quite a while. And I was overseas for 10 years, and then I came back to Australia, and I kind of fell into consulting and sort of, I guess, have had the good fortune to be able to take everything that I learned about working in, I think, one of the top global pharmaceutical companies in the world, Pfizer notwithstanding, and CSL, of course, and bring that back to the Australian environment, probably at a time when I think things were still a little bit nascent when it came to drug development. I can certainly say between now and say 10 years ago when I first started doing this, not only the appreciation of the value of something like a target product profile or the concept of doing a well controlled initial clinical study has really moved on in leaps and bounds. So in terms of the panel discussion, my contribution, I've been on kind of multiple sides of the fence. When I was at Roche, I was involved in evaluating partnering proposals. When I also worked briefly for a not-for-profit where we were engaging with pharma in getting investigator trials up. At Factor Therapeutics was an ASX listed company, so I was dealing with investors. There's a lot to talk about there, but we don't need to go into detail today. Telix, a little bit more on the kind of more like commercial collaborators, other companies setting up joint development initiatives. And now with Carpe Vitae, we have the good fortune to have been supported by a fantastic seed investor. And so another sort of angle of experience in working with a seed investor as we move towards really looking at taking the portfolio through to the next stage, which is going to then, I think, involve bringing in other investors of different kinds. And I'd be happy to talk about that a little bit more on the panel. Thanks, Mark.
Thanks very much, Ros. We'll invite all the speakers and panelists to hop up on stage. We've got time for a couple of questions. So please, online, pop them in the chat box, as well as in person up the front here. So we're all getting our lapel mics on. Okay, I might, if there's no questions in the room. First question.
Thank you. My name is Norbert Kinsler. I work with Mara Research. I thought particularly asking Anud and Adriana, I think who's still online, these days, a scientific career is not necessarily defined. You go either into pharma or industry or try the NHMRC pathway. So what are your thoughts how an early mid-career researcher can actually maybe learn some of the ropes, what a pharma wants and needs, so they actually maybe don't have to hear this, what to do or not to do, and they already know it, and which helps them with when they want to go forward with their translation research. So are there any touch points or avenues where an early mid-career researcher can come into your company and learn what how pharma thinks, what pharma wants?
Yes, that's a great question, and I'm not going to answer from my own experience because I think I've done everything in a very different way and probably not in the fastest ways in my career. I joined pharma when I was quite old and came to CSL. I was in love with academia and still am. So I think that my advice to you would be the earlier the better, because when we are in academia, we tend to think a good idea is a good product, and that's where the reality kicks in. So I think having that early understanding of what makes a good idea a potential product and a therapeutic for patients, the earlier you learn that, the better. I would have liked to have done that as soon as I finish my PhD. So when I get into the postdoc, you already start thinking about that. Good science is good science, whether it is academia or industry, so that it won't change. But my advice to you would be, if you really want to have that broad idea of how you translate and progress your good ideas beyond the patents, the papers, but into a product, the earlier you can do it, the better it will be. And there's plenty of people you can talk casually or more formally in Australia and within CSL as well.
Hi.
Oh, sorry, can I add to that?
Yeah, correct.
So is it just so keep your eyes open for any of those sort of opportunities, engaging with industry for university researchers to step into industry, just as a, for example, and if Goss was here, he'd be mentioning about the READY program. So there is a UQ postdoc who's working for a year as an intern, essentially for Brennan Capital. So that gives that person the key insight into what venture's looking for. Part of that program requires them to go back into their research role after and then spread the word. So that sort of happens. Not represented today, but another one I'm aware of is AstraZeneca, quite proactively at the moment, are seeking early to mid-career researchers. You can't be more than 5 years out from your PhD. And then they're looking at programs, and then you would have the opportunity to do some work in their labs, or indeed do all of it in their labs. And I will say that's part of the Pfizer partnership that we have through UniQuest UQ. There can be visiting scientists under the Pfizer CTI. Obviously, COVID's got in the way of that, but that is part of the ambition, and so people can have time in industry and then come back out. Lastly, as a plug, if you're from UQ, you can come to the commercialisation workshop that UniQuest runs that gives an insight into the 2 days training as to what industry is looking for.
So those are large data packages for a laboratory to produce, from discovery right through to drug PKs, okay? So if you had a situation where you had a GWAS verified novel target in diseases which currently have no medicine, and you know what the target is and you know all what it does, but you're not a drug discovery lab, what do you suggest is the option for the drug invention?
Yeah, I mean, I can take this one because I don't think you need to be a drug discovery expert. No, you have to have a molecule that is PK'd. That's just a situation where it might be ready for licensing. Someone like me at Pfizer, 99% of the collaboration are pre-drug. I mean, there's no discovery work on it, except the CRC for therapeutic deal, where there was a molecule and library was screened on the Cat6 same molecule target. None of the programs that we have funded, and that's close to $30–40 million in the last 5 years, have drug on it. But the target is novel, or the target may be novel because the scientist who's driving it is the world leader. One thing I should say very clearly, when Pfizer is looking for collaborations, we're also looking for, is this person is the one who gets invited to the top meetings in the world, right? So we do real bioanalysis on an investigator. Is this person driving it? And that gives us confidence. So I don't think you should take away the message from here that you have to have a drug discovered or discovery being done. Solid science, solid target, I'm very interested. That's the message that I wanted to give forward. So don't worry about being screening libraries and things. Just focus from Pfizer perspective or even CSL perspective, novel targets, novel mode of action. Why should Pfizer come to Australia, to be honest, or Brisbane? Then they can get a lot of information from their backyards in Kendall Square. So we need to show real differentiation. We need to show that this science is clearly Pfizer needs, because it's not there, it's here.
Yeah, I'd also say that some of us in Australia have also been at Cambridge, Scripps and other places. So I don't see that Australia is exactly a backwater of training or experience scientifically.
No, I'm not saying Australia, I mean, as I showed, there are like so many substrates. I mean, honestly, I'm so proud to have done 12, 15 very, very good collaborations, more than any other company I can claim, pharma company in Australia, even the VCs that I was calling them before, not done in therapeutic space, the collaborations that we have been able to do. So I'm not saying Australian science is bad. Australian science is so good. The frustration is that we don't translate well. And that's a different topic. But what I'm saying to academics is that science, that is, you are the driver. That's the bar, unfortunately. Previous times, you could have been an expert in TNF, and multiple collaborations could have been in the TNF area, for example. But now we're looking at competition is so much that everybody published a paper in Nature, everybody's working on it. So we're looking for pre-publications, basically. And that's the bar. So please don't take me that I'm saying Australian science. Pfizer pays me money, good money, because we are finding substrates that are high quality. So super science, no question about that. Very poor translation, and that's something that we need to change.
Thanks very much. While we take some next questions from the room, we'll go to one from online. First question here from Derek Richard. Therapeutics may have more than one indication where they may function. In this case, do you go after the largest market or go for the benefits of a rare / orphan disease? Open to the panel.
Maybe I can give CSL a go.
Yeah.
All right. I think it depends. It's case by case. CSL is focused on rare diseases, but even in rare diseases, you can have more than one that you could target with one particular drug. So the competitive landscape, the niche where that drug might make the major impact to patients, the unmet need, those are the things that we look at. So I can't give you a general answer because we have drugs that go across a broad spectrum of diseases. We have started with one indication. We might later on repurpose into another indication, but the lead indication depends on many different factors. And that's going to be pretty much case by case and where the therapeutic landscape is at the moment. If the therapeutic landscape is very well covered and we want to make big impact on patients, we might choose a different one. So yeah, it's an internal discussion on a strategy that is ongoing and change all the time.
Roz, as CEO, do you have any thoughts on it?
Wouldn't mind having a go at this one. Yeah, look, I mean, what we're talking about there is potentially the clinical development strategy or the regulatory strategy and how you ultimately plan to get your product to market in the first instance. And I think, which kind of links to another point that I wanted to make, which I think is that one of the big challenges that I see, and Carpe Vitae is working, you know, we're commercializing a technology that's come out of a lab that's based here at the TRI. And we are, in fact, developing the drugs as well as having identified novel targets. So I think it depends a little bit on to your initial question, Fiona, it depends a little bit on kind of your personal ambition as well. There are some scientists who really want to kind of get into that sort of, how do you actually make drugs? And there are, you know, there's knowledge around and there are companies that can assist with that as well. And we are collaborating with some vendors who are assisting, for example, on the medicinal chemistry side of things. So it can be done if that's your bag, but if it's not, then there are, you know, as Anand said, there are other routes. But then I think the big step that is really beneficial is early commercial input. And that's really what everybody who spoke today has really talked about, is that understanding of the patient that the drug will be used in, who's going to pay for it, how will it be prescribed, who will it be used in, what subset of patients, all of that sort of that's all the kind of commercial perspective. And when I worked at Roche, you know, we would be working on very early stage products and there was always a dedicated business person on the team who would assist the scientists with, or the team with pulling together that information. Who else is out there? How big is the, you know, what does the standard of care look like? How big is the market at the moment? All that stuff that probably feels a little bit nebulous, and where do I even begin to break that problem down? And I know, you know, obviously, Craig, your tech transfer office provides a lot of assistance with that. So, and then when it comes, and then I think part of that process also assists with Derek's question about, do you go for a niche target or a bigger indication? And it's a bit of a classic development strategy that you might actually try if you can. The thing is, I think in drug development is speed. So my answer to that would be understanding what your options are and which one gets you to where you want to be the fastest. And where you want to be the fastest is on the market with an approved drug that has characteristics that people are going to want to prescribe it and patients are going to want to take it. And that whole process of understanding what the need is and what's currently available and where the gaps are is how you really help to at least partly answer that question. Things like how long clinical trial programs take and how much they cost, it's really easy to come up with ballpark costs for those kinds of things that, you know, will neither be here nor there in the grand scheme of things if your drug is actually successful. So I think at Carpe Vitae, one of the things that we've worked very hard on with our lead asset is taking it through what we call a target product profile development. And that's, you know, a document that's maybe 5 pages long in the end, but it's actually the thinking that goes into all of that, deciding what you want ideally what you want your product to look like in the end, that has helped to sort of cement the team's focus and certainly drives a lot of the thinking now that we're getting into that sort of early research phase or non-clinical development phase.
Thanks very much, Ross. Now, I realise we've run over time, but we're really keen to keep the conversation going for a couple of minutes.
Yeah, sorry, that was a long answer.
So we've got, we'll just go to one more question online here. This one from Melody Dobrynin. To the panel, how much equity does a tech transfer office expect to take? And how much allocated equity is a red flag for a pharma company to engage with the project?
I think I'll answer
Yes,
that.
Craig. Everyone's looking at you. Craig.
There's no one answer to that. I don't think there's any red flag. It's about the partnership, the alliance. Back when I was at Cambridge Antibody Technology, it's pretty simple. You got to get alignment. You got to get all the parties aligned and realise to put 1 and 1 together to make 3. Sometimes they're non-equity deals. Sometimes they are equity deals. It's really getting the best out of everyone. I think that the Australian taxpayer does expect some returns, and we all know about the virtuous cycle. There's more medical research funding than there has been before, and then point out that. So if there are commercial returns, that is part of the return to the country. There's also jobs through manufacture. So I think it's got to be a bit more sophisticated than just looking at equity. It's looking at deal flexibility, and that's certainly something that we have at UniQuest and a lot of precedents. And just like you guys, as scientists experiment, we experiment with deal structures too. The sole motivation is get the best value out of everyone to get it to patients as soon as possible.
Thanks, Craig. Samira, did you have any insights as you were going through as a researcher, learning about all the different avenues that were available through the through the office?
I mean, I'd just like to say that I think the reason why commercialization and translation is slower in Australia is because we lack the infrastructure. We don't have the resources that are available in the UK or Europe and the US. Like, you know, for my research, I need to fatten up mice on a high-fat diet for 30 weeks. And actually in the US, you can just buy those off the shelf. So it's way quicker, which means that you get to the market easily. And I think the one thing I learned is it is going to be slower here. So you need to differentiate yourself appropriately. But also you need to be a jack of all trades. Like I had to learn how to do a budget, but keep to the budget, which is difficult. With NHMRC, we don't really do that. But with the venture capitalist company, it has to be very exact and precise. So this is like one of the things I learned very early on, and it takes a lot of time to just do the admin side of things, which I had to do myself for the first couple of years. So yeah, that takes a lot of time from research.
We're running short on time, but I know there's a question at the back from Stella.
It's kind of early engagement with pharma industry, being in EMCRs, who might have had a very excellent idea and then have something, but haven't fully de-risked the program, because that's one of the requirements that you should be proposing to. But then by the time the EMCR try to do it, it might be a bit late. So is pharma company or kind of a sort of education system that provide us or help us to find the way to have some initial funding or some resources to de-risk this, these early challenges, so that you can engage with pharma, a bit more solid way.
I'm not sure I can answer that. Look, when I was in the lab, it used to be Friday afternoon experiments.
Yeah.
That was the time to try out your own ideas, generate some data, convince the prof that this could have some tractability, could be viable science. I'm actually going to throw it open to others.
I think the other thing is that I don't know, some universities, at least, I'm not sure why they do, how much is it, but some university have a system where they provide $30,000, $50,000 grant to do an animal experiment that could be useful for validation, for example. I know University of Melbourne does that. So that means that you have some work that you could do with that amount of money, and then come back to or go back to the company with that data. I can tell you that I have actually written a letter of support for University of Melbourne investigator who was being shortlisted for that kind of $30,000-$40,000 grant from the university. Not saying that Pfizer will take it, but because I had evaluated it, there was some feedback, and not just Pfizer, but other companies also giving the same feedback. Then I wrote, yeah, that experiment will be critical for us to reevaluate the possibility of working with that investigator. No guarantee, but that could happen.
I mean, I'd just like to add to that. We still haven't de-risked our therapeutic completely, and we got the venture capitalist funds beforehand because of our strong patent position, but also because it can be differentiated and there's a huge unmet need. So I think differentiating yourself in the other aspects and then pitching the de-risking within your program of research that you're going to do within the development plan is a good idea as well. So as long as you have a strong stand along the patent position, I think you're good.
I think, unfortunately, we've run out of time. So
There's one question here in the room. Surely we can do one room.
I can speak really loudly. So I'm just wondering whether actually one of the first barriers to getting into tech transfer company at your institution on board. I'm not sure about your experience, Samara, but that actually made a massive difference to myself to actually have UniQuest starting to help guide you through some of those more complex aspects of the data pack that an academic doesn't usually work on. And so, you know, is the resourcing in UniQuest sufficient? Because I find I have conversations with colleagues and actually getting past that first barrier of getting UniQuest attention is actually quite a significant one as well. So I'm just wondering whether, Craig, you can explain what you're really looking for and how much earlier in the pipeline, because you're suggesting that we should be thinking right from our early phase discovery, what our endpoint is, how early in the pipeline should we be engaging with UniQuest, and whether you've got the resources to really support a large amount of translation from University of Queensland.
Thank you. So certainly that is what UniQuest is there doing and wants to continue to do,
Yeah,
and makes itself available. It is, there's obviously more researchers than there are at UniQuest, but actual fact, we are looking for those early stage so that we can help guide, particularly when it is early stage research, to put in the right directions or the right comparators, standard of care. Happy to chat with you. I think it is important to have connections with people in UniQuest and find that. For others, it's important to have connections with your tech transfer office. It can make a world of difference. You don't get another shot at grant funding. So if you can do the extra bit of experiment with that grant funding that makes it interesting to the company, that's what we want to do. In terms of conversations, there's certainly people, we have people all around campus. So if they haven't managed to find time with you.
But they're still finding that getting even UniQuest on board can be a challenge. And so Samara has been successful at that, and then UniQuest were right on board, so they helped her even change her disease focus, etc. I know her story well. I've been working with UniQuest, they've been absolutely great, but we've had others who have ended up translating their research that had barriers with actually getting UniQuest on board, and others who are even earlier phase who are really challenged, finding it that they're not getting much return from their engagement with UniQuest. So it'd be really helpful to know what they need to change in their conversation with you to get you on board, because I think it does make a huge difference once you're backing a project.
Yeah, look, thank you for saying that. Really pleased to hear it, and we definitely make ourselves available. So if it's not happening enough, you know, chat with me. We're all there to help move things forward. And if there is more sort of like self-guided as well, but we actually want to work together. It's a team sport.
I'd just like to add a couple of lines to that. I mean, I think UniQuest's approach is conservative, which is a little bit of a challenge for an academic to pitch it in the right way. And I think when we found exactly how to pitch it to UniQuest, they were straight away on board. But I think is that communication is a little bit different to our pitches for grants. So I think that's the, you know,
That's
like
what I'm trying to get at. What's that magic formula that changes how we should be changing our pitch to get your attention?
Yeah, look, and just it's the line of sight of the product. So it's not the science per se. It's that line of sight of product. And you don't necessarily have to have that in the first place, but that's what it's getting towards. And that's why it is an easier process. We'd like to know early, and that's where there can be a bit of steering. There might be a better disease indication, might come back to your initial one, if that's your heartland, when the first has been successful, there's more commercial pull. So it is conversation.
And I can just say one word on this one as well, because Craig and the team at UniQuest, for example, facing us with wish lists so demanding that we need this totally novel. So they have to be quite tough in a way, because they don't want to pitch that may not go down very well. So they had to be very selective, I think, as well.
Yeah, completely
And they do
understandable.
fantastic. I must admit that, you know, UniQuest is one of the best organizations.
But I think a lot of us don't approach UniQuest with the right type of information as well. So that's what I'm sort of trying to get at.
And from time to time, there are specific call proposals for many different companies on topics, which UniQuest supplies to all of you. So that's another way of entering, not bypassing UniQuest, but what I'm saying there, you can say, well, this is the space J&J Pfizer is looking for at the moment, then you can go and see what that could fit into.
Awesome, thank you.
Good points. Yeah, and we like that conversation circle.
Conversation keeps going for a fair bit longer, but I think we should leave it there. So I just want to thank you all for attending online and in person, and especially thank you to all our speakers in person and also online. So leave it there. Thanks again for joining us, and keep in touch for our next seminar coming up. Thanks very much.